because there's so much to learn. Okay, so let's take a look at where we left off with our synthesis. We had talked about the synthesis of amino acids. So now we're going to be looking at the synthesis of nucleotides. All right. And there's a couple different ways we can do this. Just like with our proteins, we can either make them from scratch or we can recycle them from proteins that have been broken down. So same thing with nucleotides, we can synthesize them de novo, or we can salvage them from RNA, DNA, and cofactor degradation as well too. Okay. One of the neat things we see is that a lot of parasites can't synthesize them. They lack that de novo biosynthesis pathway. So we can potentially target them by developing drugs that inhibit the salvage pathways. All right. And we'll go over this in a little bit. Okay. So as we start to make these nucleotides, our salvage pathways we will talk about here in a little bit, but let's talk about the de novo synthesis first, because this next picture is going to be awesome because it's so lovely and complex as to where all the components come from. Okay. The neat thing is the de novo biosynthesis pathway is pretty much the same across organisms. So that means it's probably a pretty important pathway here. The bases are synthesized while they're attached to the ribose. So we start with a ribose biomolecule, or at least a ribose-related molecule. Most of the amino groups are going to come from glutamate. Glycine is a precursor for the purines, and aspartate is a precursor for the pyrimidines. Our nucleotide pools are generally kept fairly low, so the cells must continuously work to make them to maintain that homeostasis. We don't want to have a lot of extra nucleotides floating around out there. Okay, and the neat thing about this is that because these pools are low and this constant metabolism is occurring, that may actually help limit rates of transcription and replication within the body. So we have, maybe a concentration dependent form of regulation. Ooh, but that's, not necessarily known to be true. All right. So let's look at our purines. And, for me, I always found it weird that purines, purine and pyrimidine, purine is a shorter word, but it's the bigger molecule. So that's how I remember that. So purines are the double ring structure here. Where do these different atoms come from? Well, one of the carbons comes from carbon dioxide, two of the carbons come from formate, two of the carbons and one of the nitrogens come from glycine. You get another nitrogen from aspartate, and then you get the remaining two nitrogens from glutamine. All right, so there's a lot that's going into making this, right? So we can kind of track these down here. It might be a fun thought experiment to figure out, well, how do you know where these are coming from? And this is where you could use, radio-labeled compounds. Suppose you made, a glycine with radioactive carbon 14 or something like that. Then you could, take a look at your product in here and your different components and figure out where, where those carbons were going. Okay, and at what step. All right. So don't need to memorize all this, but you should be able to say, well, hey, what happens if we alter some of this stuff? You need to understand it, but not memorize it. We're getting away from memorization at this point. Oh yeah.
So are you saying, like, if there was a reduction in glycine, for example, in the body, are you saying that the purine rings wouldn't form, or they'd find another way?
So I don't think you'd ever get to the point where glycine would be so low that you wouldn't see formation. If at that point, you're probably at starvation and close to death. So, but you could track where these are coming from. So suppose you had aspartate with a radioactive amide group in it. You would see that nitrogen would be incorporated in a 6-membered ring. You would never find it in one of these one of these positions over here in the 5-membered ring. Same way, if you had radioactive glutamine, you'd see that the amides, the nitrogens would go to one to each of the rings in there. And that's actually, the way this was discovered was by using carbon-14 and nitrogen-15. All right. So all synthesis of our purines begins with our PRPP that we mentioned last time. That's our phosphoribosyl pyrophosphate. Okay. So we start with that lovely little ring structure in here, and here's a picture of it down here. You can see it's got these phosphate groups on the different sides that help activate it. Okay. Um, so we start with PRPP, and we're going to build our ring, by adding our glycine. Okay, and then the first thing we're going to make is inosine or inosine monophosphate, actually. So how do we make this? Okay, so here we have our PRPP. We're going to take our glutamine to add this nitrogen group over here. Okay, all right, and then we add our glycine onto this R group up here. Okay, so we've attached it to the ring structure. We make this glycinamide, and we have this informal folate that's going to start adding more stuff in here. And we're going to see this continue on through. Okay, so we're building this attached to the ribose group. Okay, so now we start adding more and more and more and more. So eventually we have something that looks kind of like this, and you can see where this 6-ring structure is going to be coming from. We're going to close this ring here, and one of the next steps, actually not immediately, we need to do a little bit of reduction first in there, and so we end up making something that looks like this, and this is our inosate monophosphate. Okay, so our PRPP, we basically take that diphosphate group off. We start by adding nitrogen there, and then we add the glycine into this as well too, and start building onto that from these different components in here. Let's see if I see where our carbon dioxide is coming from. Just didn't click back far enough, right? Yeah, so you can see our carbon dioxide is coming in here from the bicarbonate. Okay, and you can see we're spending some ATP to do this. So it costs energy to make our nucleotides. All right, but ultimately we get this inosate IMP monophosphate in here, and then we can modify this to make either adenosine or guanosine. I guess I should say adenylate or guanylate because it will be phosphorylated. Okay. All right, so you can see we start here with our purine that's attached to the ribose. We have a 2-step process to get to the adenylate, where we basically kind of change this oxygen here, add an aspartate to replace that, and then cleave off the carbons as fumarate to give us our adenosine. If we go this way, what we're going to do is we're going to end up adding an oxygen here and then replacing that with an amide group to make our guanosine. Okay.
Those two are part of the ring, or they're two separate?
So at this point, our inosinate is the complete ring structure, the complete purine ring structure. What we're going to do to make adenine, we need to get rid of this oxygen and put an amide group there. So whoops, come back. There's a 2-step process to do that. So we're not really changing the ring structure. We're just changing one of the substituents that's on the ring structure. The same thing down here, if we go to guanosine, we make this xanthylate. Okay, where we actually add a carbonyl oxygen attached to one of the ring carbons here, and then we replace that oxygen with an amide group. All right. Okay, so key takeaways from this. We start with phosphoribosyl pyrophosphate. We build on that ribosyl group there to build up our purine, and then we make inosinate, and then inosinate can either make adenylate or guanylate. Okay, so that's the takeaway from that. Okay, so if we look at regulation, let's start simple and look at it in a bacteria. If we look at E. coli, there's a whole bunch of feedback inhibition that occurs here. So if we take a look at this, you can see our ADP levels. So that's basically adenylate Whenever you have ATP or GMP or an phosphate, it's going to feed back and inhibit these enzymes here, whether it's the PRPP synthetase to make PRPP. If we have lots of ADP, that's all well and good. If we have lots of AMP, GMP, or IMP, it'll feed back to that first step of the glutamine PRP aminotransferase, where you add that nitrogen, that amide group to the PRPP and replace the two phosphates in there. Okay. You got a whole bunch of steps there that ultimately will lead to IMP. All those are going to proceed. So if we have lots of 5-phosphoribosylamine, then we're going to start making our IMP. The neat thing is if we have lots of AMP, it'll feed back and inhibit this pathway. If we have lots of GMP, it'll feed back and inhibit the other fork in there. Okay. All right. So our end products are doing the inhibition, and that's what we say about that. All right. So I'll pause for just a second there. Yeah, this is E. coli. It's, I think, similar in mammals, but anyway. But that's our purines. Okay. So start with PRPP, build on the ring, get an ozonate, and then split to either AMP or GMP. If we look at pyrimidines, okay, here we have a completely different mechanism. All right. So first, we're going to make the pyrimidine ring, and once we've made it, we'll attach it to the ribose group. Okay. So we don't use PRPP to make this. So what happens? Our first step is we're going to combine our aspartate and our carbamoyl phosphate. Oh, there we go. Remember that from the urea cycle, right? And we're going to combine those two to make something called orotate. Okay. I don't know if you've seen, which professor is it? Somebody's got a license plate that says orotate out there. I guess I'm assuming that's what they study.
What does it say?
Or orotate, this compound. Yeah. So it's the most important compound in the world for them at least. Yeah. All right, so we make our orotate, and here we have that lovely little 6-membered ring. Okay. Then once we have that ring, we will attach the orotate to our PRPP. Okay. Okay. So we make the ring structure, attach it to ribose, and then we're going to modify the base to make either cytidine or thymine or uracil, I guess. Okay. But I guess first we're going to make uridylate there. Okay. All right. So we make uridine first. Okay. And then we can phosphorylate that to UTP, and then we could convert that to CTP. Okay. All righty. Okay. So not too bad, right? So I guess you'd say, you actually, and this is one of these kind of neat things where, as opposed to making like in pyrimidine synthesis where you have the IMP, which can either make your end product of GMP or AMP, here with purines, you make UMP, and then you can have another reaction to make CMP. Okay. Or sorry, CTP. Yeah. So it's not a split, it's just a continuation or discontinuation. All right. So how do we regulate this one? Well, we again see feedback inhibition in here. So if we have lots of CTP, higher concentration of CTP, we will inhibit this reaction. Okay. On the other hand, if we have lots of ATP, so we have more pyrimidines, more purines, then we're going to stimulate our pyrimidine synthesis. Okay.
Why is there more ATP when there's more purine? Is that what you're essentially saying?
So I'm saying if you have more ATP, you're going to make more of the pyrimidines.
Oh, okay.
Okay, yeah. Sorry. Yeah. There too. Okay. All right. And that's really kind of neat to see if you have, relatively high concentrations of CTP and ATP, it's still going to counteract that feed. Basically, ATP is going to counteract the feedback inhibition. All right. So all these are our ribonucleotides. So we've got AMP, GMP, UTP and CTP, I guess, do the triphosphates in there. These ribonucleotides are the precursors to the deoxyribonucleotides. Okay, so we're going to directly reduce this hydroxyl group to a simple carbon-hydrogen bond without activating the carbon. So here we need this kind of neat. I mean, this is a tough reaction. This is not something that you would normally see happen spontaneously. So we have this very specialized ribonucleotide reductase. Okay, and we're going to see that 2 protons are donated by NADPH, and they're carried by thioredoxin or glutoredoxin to help in this reduction. All right, so there's a lot of mechanistic stuff on these next few slides, and I don't know. But the key is we start with NADPH that donates that, and then we either go through glutathione or thioredoxin to get there. And you can see our sulfur groups are being oxidated or reduced as we go along the pathways. But ultimately, we get there and we donate these 2 protons, and we remove our hydroxyl group to make water and replace it with the other hydrogen in there. Okay. So you can see our structure. We have a bunch of those sulfhydryl groups. Oof, man. All right. The mechanism is actually really kind of neat. We are creating a stable radical in there. Okay. All right. We can look at this, in part because we have this lovely little ring structure in here, but I'm not going to ask you guys to memorize or go through the details of this radical formation. But I do want you to know that ribonucleotide reductase, what it does, it makes our deoxy form of our nucleic acids, and it has this kind of unique mechanism. Okay? And of course, we'll talk about its regulation as well too. Okay. Our ribonucleotide reductase has 2 types of regulatory sites. One of them is going to affect the activity. So if it binds ATP, it's going to activate this. Okay. On the other hand, if we have deoxy ATP, it will inhibit it. So if we have higher amounts of ATP, we can say, we'll make more of the deoxy form. If we have higher amounts of the deoxy form of adenine triphosphate, then we're going to say, slow down, we got enough. Okay. But there's another site in there that actually affects the substrate specificity. Okay. So if we have high amounts of ATP or DATP, either one, then what we see is ribonucleotide reductase will have less specificity for adenine and more specificity for UDP and CDP. Okay. So we see this oligomerization that occurs to balance this. So basically, if we have an excess of one type of nucleotide, we are going to inhibit the making of the deoxy form of that and promote the making of the other ones. So we want to basically try and keep a balanced ratio of our various deoxy nucleotides in there. So you could say, what do we see here? All right, so ATP promotes the formation of the deoxy form of all of them, okay? DATP will actually feed back and inhibit this at the one site, okay? But here we can see, what we gonna do here? Our DGTP will actually promote, the formation of DATP, or so go over here. Right? Yeah. Okay. They can inhibit DTTP and DCTP. And this is kind of feedback inhibition here. And you can see this is going to promote across this way. So you can see our purines, okay, are going to inhibit the formation of other deoxypurines and promote the formation of deoxypyrimidines and vice versa. Okay. I know that's a tough concept. I can see. Take a second to think about that, right? Okay. So if I have more of my purines in the deoxy form, they're going to say, no, we got lots of purines, go make deoxypyrimidines. If I have lots of deoxypyrimidines, they're going to kind of feedback and prevent making more of those, but they're also going to promote making more of the deoxypurines. Okay. It's like, I never feel like this picture does justice to that, but I think that's a better way of saying that.
But this only involves the deoxy form?
Right, yes, yeah. And that actually leads us into, well, wait a minute, we don't have a deoxy UTP, except as an intermediate. So we need our thymine to make our deoxythymine monophosphate. We actually first make a deoxy form of UTP and then convert that to TMP. Okay, so we make a deoxy UTP. That gets dephosphorylated. We cleave off pyrophosphate by that deoxy UTPase in there. And once we have dump, we convert that to deoxythymidylate monophosphate through this thymidylate synthase. And it's going to add a methyl group from our tetrahydrofolate that we mentioned before as a good single carbon transfer unit. All right. And then our thymidylate synthase makes a good target for some anti-cancer drugs because, of course, cancer cells are rapidly dividing and growing, so they need lots of nucleotides. So if we can inhibit the formation of TMP, we can inhibit the growth of the cells in there. And of course, that's any fast-growing cell would be targeted by that. All right. Okay, so here is a short version of what we've got going on here. All right. So a ribonucleotide reductase can make the deoxy form of either one of these. Our kinase will then convert it into the triphosphate form. We can make our DCTP, we can deaminate that to DUTP. Our DUTPase can make this monophosphate form, which thymidylate synthase can then make the thymine. All right. And this just shows the mechanism of how this occurs. And again, we need a tetrahydrofolate in here. Okay. So let's talk about the tetrahydrofolate a little bit as well too. If you have a deficiency in folic acid, that will lead to reduced thymidylate synthesis. Okay. So if you have poor nutrition, if you eat at McDonald's every day and don't eat your leafy green vegetables and stuff like that. So any type of, I always talk about this because it surprised me moving down to Winston-Salem because I come from West Virginia and we're all poor country bumpkins up there. Okay, not really, but that's kind of the stereotype. But Winston-Salem is one of the most food insecure regions in the country. There's a stunning amount of food insecurity in this little area, which is, several organizations have been working to address that, but it's still a pretty significant issue. So, there's a lot of people who just don't get the proper nutrition. So if we lack folic acid in the diet, then we basically get uracil that's incorporated in DNA. Repair mechanisms are going to remove that, but by removing it, they end up causing strand breaks in here, and you can see an increased risk of cancer, heart disease, neurological impairment. If you think also of tetrahydrofolate in pregnancy being critical for neural tube, neural crest formation as well too, that may be related to this neurological impairment as well. So, and again, that's also why pretty much anytime you go to a neonatal doctor, the first thing the OBGYN will do is hand you a big bottle of folic acid supplements, okay, just to make sure that you're getting the folic acid in there. So make sure, basically without this cofactor, we can't make thymidylate, right, which is critical for healthy function.
I learned in my nutrition class that neural tube defect
Uh-huh.
actually happens like in the first 6 weeks.
Yes.
So
Yeah.
actually, they say all women of childbearing years should be taking folic acid because
Yeah. Yeah.
usually you don't even know you're pregnant by
Yep.
the time you've already caused damage.
Yeah. Yeah. And it's just, so that's why leafy greens are good. Folic acid in there, or any supplements in there too. And it's just, I always look at too, like at childbirth and pregnancy. It's like, on the one hand, studying some of the mechanisms that are in there, it's amazing anyone ever gets pregnant and has kids. And then on the other hand, it's like, it's amazing people don't get pregnant more often. And so it's just, whew, it's a reason to study statistics, because there's, it's crazy. I remember when I was first learning developmental biochemistry, and a lot of it is just, well, how do we develop into mesoderm, endoderm, ectoderm? It just depends on the diffusion of a few cells that release some factors, and then is it VEGF and various other growth factors in there, and the different concentrations lead to the differentiation of those cells. And it's just simple diffusion. So you've got the statistical measurement in there too. So
I've never understood that either. They say all these factors have to be aligned to get pregnant,
yeah,
but then you have 15 and 16-year-olds going out getting pregnant like after 2 or 3 times. Like
Yeah.
I just don't get that at all.
yeah, it's just there's a lot of statistical, I don't want to say luck, but statistical occurrences. Yeah. If the stats line up, and it's surprising. It just seems like, it'll never happen. It'll always happen. So yeah. Okay. So that's how we make them. How do we chew them up? If we catabolize our purines and our pyrimidines, we get different components in here. So one of the things that happens with purines, okay, so we got our big ring structures there too. We will have these nucleotidases that will kind of cleave off the ribosyl groups, and we'll start deaminating them as well too. And we end up making xanthine, okay? So you can see here our guanosine, and at least guanine. We end up making xanthine there too. Our AMP, we get adenosine back to a nosine, back to this hypoxanthine in the keto form. So we get to this xanthine. And then our xanthine can be converted to uric acid by xanthine oxidase. Okay. All right. It's really kind of neat because spiders and other arachnids lack xanthine oxidase. Okay, anyway. But our xanthine oxidase can create uric acid. Okay. So uric acid then can get converted into allantoin, allantoate, and urea, blah, blah, so on and so forth. And we can see our various forms of excretion that'll happen here. So what happens if we get excess uric acid? You get the gout. All right. And there's a picture. I don't think I have it in here. I need to insert it into these slides. But if excess uric acid will actually crystallize into a solid form, often in the joints, usually in the extremities, and these uric acid crystals, cause a very painful condition. All right, and this is for some reason primarily affects males. I don't know. It can be treated by avoiding purine-rich foods like seafood and liver and alcohol, or avoiding fructose as well too, which is important in there. It's kind of neat because gout was once known as the rich man's disease because you had to eat a lot of rich foods and be able to afford to eat a lot of these rich foods. And overindulgence could lead to the formation of gout. And the picture I have is like the little demon biting somebody's toe, because apparently gout is very, very painful. There's also another treatment. You can use xanthine oxidase inhibitors like allopurinol, and they will prevent, yeah, that xanthine oxidase from converting xanthine into uric acid. So this leads me to like the old wives' tale, right? You should. What happens if you have gout? Eat lots of cherries. Why? Cherries are high in allopurinol, and they'll inhibit xanthine oxidase. So there's actually some logic to that. So, or was it? I often wonder too, because I see like tart cherry tart cherry juice sold in grocery stores. I was like, huh, is that, I haven't been in like a
My
health
dad
food
had a
store.
really bad gout, and
Yeah.
he drank that cherry juice all the time.
Okay. Yeah.
Yeah, it
Didn't
didn't really help a lot.
didn't help a
but
lot. Yeah.
he
Yeah.
felt maybe it was almost like a. I think that cherry juice has like so much sugar in it
Yeah. Yeah.
that it
The,
kind of like,
the fructose may have been overwhelming the because
yeah,
so it'd probably better to actually just eat cherries
yeah,
than drinking the cherry juice there too, and of course avoiding other foods too, like stay away from the liver and the booze and all the other good stuff out there too. Liver. Got to tell you my story about liver. So growing up on a farm, we butcher cattle and have all sorts of meat in there. And I'd say I was at college, so and here's a big cooler. I remember it was funny. I lived in a house with a group of people, and we all brought back coolers full of meat one time, because we're all country kids, whatever. But one time I was getting food out of the freezer, and I tried to mix it up and get a variety of things, mostly hamburger and whatnot, some steaks and stuff. But one time I accidentally grabbed some liver. And I was frying. I was like, well, I'll fry liver. I was like, huh. And I had a roommate, and we were eating together too. And we both took like one bite of liver, and I was like, this is really bad. So threw it out and ordered pizza. Oh, it was terrible. I'm not a liver fan.
I used to love liver.
Yeah. I say the only time I've eaten liver was as a pate. There's like a Braunschweiger spread that had a significant amount of ground liver in it, or spreadable liver or whatever. But you put that on crackers with some cheese or something like that, and it was okay. But
my mom used to put liver in her stuffing. It was disgusting.
Not something I can, get behind. Okay, so our allopurinol inhibits xanthine oxidase, so that we won't make our various stuff in here. If we look at our pyrimidines, on the other hand, what we see is our catabolism in here. Okay, random stuff here. We will ultimately get ammonia and urea. We can also produce some various intermediates of the citric acid cycle. So thymine can get degraded to succinyl CoA, and that can feed directly into the citric acid cycle in there. So usually pyrimidines, you're not going to see any crazy diet-associated diseases like gout with those. It's the purines where you get that from. All right. So we can also recycle these. We have salvage pathways as well too. So we're going to freebase. Here's where I'm going to make what was his name? Famous comedian. Richard Pryor joke about freebasing. And catching on fire. That's way before your time, but yeah. Okay. But our free bases get released, so they're no longer attached to the glycine in there. They get burned off. Okay, never mind. Terrible. I'm a horrible person there too. Yeah. But they get released and they can be reused. So for example, we could take our adenine and react it with PRPP to make AMP. Okay. One of the neat things is that the brain is really dependent on salvage pathways. So we'll see this. If we lack some of these enzymes that are involved in that, in particular, if you look at hypoxanthine guanine phosphoribosyltransferase transferase, if that is missing or defective, you get Lesch-Nyhan syndrome. And this is an absolutely horrifying disease because basically you have these little kids who can't salvage this, so they get a buildup of these free bases in their brain, and you see severe neurological impairment. And one of the things that this manifests as is they tend to bite their fingers and toes, and they will actually chew off their fingers and toes. So the treatment here is, we'll avoid these. I guess dietary treatment here. It's not a good thing. It's a bad thing. And we'll talk more about Lesch-Nyhan syndrome later on as well too.
That
hypoxanthine guanine
transferase.
Yeah.
Is that one of the
that's one of the salvage enzymes in there. Okay. There are a lot of pharmaceuticals that target these pathways, either the biosynthesis pathway in particular. A lot of these can be used as chemo drugs in there. So we can have glutamine analogs. Our glutamine analogs are going to inhibit the glutamine aminotransferases. So keep in mind our glutamine is providing nitrogen to our purines. So if we have these analogs, they're going to inhibit that, and that's going to slow the growth of the cancer cells. We also have this fluorouracil. So fluorouracil will get converted by the salvage pathway into this fluorinated deoxy-UMP, which will inhibit the thymidylate synthase. It basically just, you can't convert, that fluorine group in there prevents the function of the synthase in there. Methotrexate, which is prescribed for several things, I think, and aminoperidone are actually going to inhibit the dihydrofolate reductase. So they are competitive inhibitors of that as well. And that will, again, work to slow cell growth in here. You can also look at various antibiotics. So this is where you get into, looking at the different enzyme forms between species. So if we look at an antibiotic to treat a bacterial infection, our trypanosomes you can possibly treat with allopurinol because those trypanosomes lack the de novo nucleotide synthesis pathway. So they have to get their nucleotides from the salvage pathways. So if you inhibit salvage pathways by, say, like using our allopurinol, then you can't, they presumably can't, they'll die because they can't incorporate the right nucleotides. Trimethoprim is going to inhibit bacterial dihydrofolate reductase. But here's a neat thing, it actually binds very strongly to the bacterial forms of it, but doesn't bind as strongly to the human forms. So we can tolerate it. It's gonna marginally slow down. It's like, when you see a cop on the interstate and you go from like 80 to 75, okay, it's a little bit of slowdown here, but it greatly inhibits the bacterial salvage pathways. Okay, so that's this chapter, and oh my God, that is a lot in this chapter. Massive. The good news is this is probably the worst chapter. I don't know, maybe 23. We'll see here in a second. But what I'm going to do now is actually just kind of pause my recording here, and we're going to talk about the exam a little bit. Okay, so now I need to click off of this. Okay, I need to find my exam, and I did a horrible thing when I made my exam. As I was updating my exam, I changed I changed my answer key. I lost my answer key. It was terrible. Okay, sing this down here. Move this out of the way. I can find the things that I want. Okay. This is exam 2A2, right? All right, okay. So for the citric acid cycle stuff, so you guys can kind of pretty much, just look that up on one of the things there. That's one of those horrible memorization questions in there, and it helped if I actually open the folder, wouldn't it? Come on. There we go. Okay, so come on. All right. If we look at the pyruvate dehydrogenase complex, remember it has 5 cofactors. These are the TPP, the FAD, that lipoyl lysine, NAD+, and coenzyme A. Okay, and that's that trifunctional enzyme that's involved in the first step for our lipid synthesis. Okay. As far as these cofactors in here, what do they have in common? They all transfer methyl groups, single carbon units in here. So etomet, biotin, pyridoxal phosphate, or tetrahydrofolate, they all transfer 1. And then as things that they have that they differ in, I mean, that could, that was pretty wide open in there. For example, if you picked like, was it etomet and biotin, you could say that etomet is 1,000 times more, more specific for this, and biotin is a little more, not as good as etomet as doing that transfer, but it is, more widely available to other compounds or something like that. Anaplerotic reactions replenish intermediates of a metabolic pathway, and they do tend to be regulated. The urea cycle is eliminating, and somebody said urea, and I was like, well, okay, that's technically correct, but we're really looking at ammonia in there as well. The first compound that gets formed is carbamoyl phosphate. Carbamoyl phosphate synthetase 1 is a compound that makes that. How can it be regulated? We could look at enzyme expression levels, or there's also that N-acetyl glutamate that will bind there as well. Okay. My tough question here was about the metabolites of the central metabolic pathway. Both isoleucine and phenylalanine can be metabolized to more than one metabolite that's in there. Transamination, the first thing is our pyridoxal phosphate. Okay. Ketone bodies are made in the liver, and I realize this is probably a horrible page because almost all the answers are D, but it's just the way it worked out because I put them in alphabetical order. Free fatty acids with odd numbers. The last pass leaves 3 carbons there, so that's propionyl CoA that gets made last. So there is a slide that actually kind of shows the connection of these two, and ornithine is the only one that is not part of the connection between urea and the Krebs cycle, the TCA cycle. Ornithine is exclusive to the urea cycle. And then finally, where do our branch chain amino acid degradation occur? Every place but the liver. So the liver is the correct answer twice. It's because the liver is the most important organ out there. Anyway, those are the shorter ones. Let's do this one here. Okay. For my longer questions, okay. Actually, for the open book part, I was very pleased with the class as a whole. You guys did a good job on the open book part. So our vitamin B12 is unique due to this core and ring system. One aspect that makes it unique is it includes cobalt. So it's got that cobalamin in there as part of its name. Where do we get it from? From our diet. Okay, we have to eat particularly things in there, and there was actually, a lot of good insight into that. I got more than I necessarily wanted there, but that was, I think you guys did a good job on that one. And then the deficiency of vitamin B12, I may need to change this answer up because people are finding intrinsic factor. You guys are doing really good on that as well too. There's a compound called intrinsic factor. I think it might even be like IF1 or IF2. I forget in there, but that's critical for the formation of vitamin B12. For our fatty acid catabolism question here, again, this is pretty good. Our hormone-sensitive lipase cleaves triacylglycerols into free fatty acids and glycerol. Free fatty acid 18,2,9,12. You guys were able to find the name of this as well too. How does this fatty acid get into the bloodstream? That acylcarnitine transporter. That would be correct because this is a fairly long one. As opposed to that, you'd have to kind of look up capric acid. Capric acid is a relatively short, I think it's either maybe a 6 or 8 carbon fatty acid from milk. It is a, oh, sorry. Uh-oh. Oh, they might not be in order. I think they're out of order here. Yeah. Because my key is, yeah. It's on a different page. Yeah.
That's number 4.
Yeah. But our capric acid is a short fatty acid, so it can freely diffuse across the membrane to get in there. And then everybody actually did a good job of this. I think the answer ended up being like 117, 115, 117. There was some variability in there, but everybody was within the closeness that was in there. I did a good job of explaining that. For the D amino acids, everybody actually did a good job on that one as well too. Those are consumed, and then in some cases they could be racemized into the L form to be incorporated, or they could simply just be metabolized for energy as well. But the D form won't be incorporated into proteins, but you can racemize them to the L form. For the liver question here, alanine, both ALT and AST levels tend to be increased. From somebody with viral hepatitis. Viral hepatitis is going to cause damage to the liver. When there's damage to the liver, it's going to release components from the damaged cells into the bloodstream. So we're actually kind of, basically killing liver cells. So our ALT and our AST are going to be released in there. I can't remember if this year or previous year, somebody went way, somebody clearly was doing some medical shadowing because they were talking about the difference in levels in ALT and AST and the time frame. So there's but I was like, no, we don't need to go that far. And then my last question here is my awesome question that I was very happy with. And it was meant to be more challenging, but again, you guys did a pretty good job with this overall. Our isovaleric acidemia is a genetic disease where we are missing a particular enzyme in here, and people end up smelling like sweaty feet. Nothing like a question about, hey, why do you smell like sweaty feet? All right. So what happens is our leucine gets converted into some intermediates here, and isovaleric-CoA would normally be converted into either acetoacetic acid or acetyl-CoA, but without that enzyme, they end up getting shunted into isovaleric acid, hydroxyisovalerate, or isovaryl glycine. Okay, so because of the type of disease this is, you would typically see, in extreme cases, was it coma, tremor, whatever our neurological effects are, kind of all of these amino acid catabolism diseases have a particular defect, have similar symptoms. Sorry, in there. Roughly half the cases become apparent shortly after birth, and then the others come on sometime later in childhood. So this question as to why there might be two distinct periods requires you to kind of think about that. Well, what's going on? And you might look at two different isoforms, or you might look at homozygous versus heterozygous. Either of those could work there. Okay. What would you expect to see if you completely eliminated leucine from the patient's diet? This is a tough part of this question here too. Right. On the one hand, if you completely eliminated leucine, you would not be making the isovaleric acid, so it would not build up in the brain. On the other hand, so that works. Sounds like a good treatment, right? Except leucine's an essential amino acid, right? So if you completely eliminate leucine from the diet, then the patient's going to die from lack of leucine. So yeah, so we can't completely get rid of it. We have to maybe balance it in there. They do say one treatment during acute episodes is to give glycine. Why would that be helpful? Well, the disease is caused by a buildup of IVA, okay? But you can see that isovaleric-CoA can actually make three different compounds in here. So if we add glycine, then we may promote the formation of this isovaryl glycine, which does not have the same deleterious effects as the isovaleric acid. So we make an intermediate that is less damaging to the body. And then, of course, a treatment plan, limit, leucine, or, there are a variety of things that are in there. And I've got some links in here. So it's often similar to, basically monitoring the diet. Okay. Whew, good times, right? So, but I like this set of questions about isovaleric acid because there's a lot to suss out in there, and there's some things that are more obvious than others. There's reading the pathways. There's recalling that we can't just get rid of leucine completely. We can think about this. So I like this kind of integrated question.
I love these type of questions, even though they're a lot harder. Like that's the kind of stuff that I'm gonna, through diagnosis, that's the kind of stuff that I'm gonna be encountering. So.
Yeah. Yep. And for the upcoming take-home exam, you'll see more types of questions that'll be kind of like this, where you'll have to go figure stuff out. Okay, so I'm gonna try and do something. try and do something unique with my exam. Probably 3D. Oh, it's in 3D. Wadded up the third at you. Okay. Oh my, that's terrible.
A question about how does the method in the fatty acid differ with common milk?
Yes.
What is, you put like small chain diffusion, What does that mean?
Yes. So there are three mechanisms to get fatty acids into the cell. Long chain ones require that acyl-carnitine transporter. Medium chains one have a different one, and then the small chain ones will just diffuse across. Yeah. Okay. All right. So now, all right. Gotta make sure I can check my time here. All right, good. So, on the one hand, I kind of feel bad for you guys because it is a slightly shortened semester. On the other hand, still got 6 more chapters here. We are definitely not going to have the exam ready on Monday, maybe Wednesday, but I'll probably give you a take-home exam the following Monday. So I think that'll be the 26th. But we'll be covering new material in between there. But let's take a look at this next chapter. And I think the chapter we just finished up is one of the most dense and complex ones. And I'm never sure how students look at this chapter, whether it's a relief because this is the summation of the entire semester so far in here with our hormonal regulation, whether it's nice that it kind of tries to tie everything up, or whether it's the worst chapter ever because now you've got everything we've learned this semester all interacting together. And I'm so proud of one of my questions that I have. So let me unpause this thing. Come here. Where are we at? Did I sign out of the Zoom? Did I? I don't think I did. No. Come back here. Come back here. Damn it. Maybe I did. Yeah, whatever.